micro-robots could be used to deliver drugs directly to brain tumors, offering new hope for treating challenging neurological diseases. In a recent study, scientists created magnetically controlled micro-robots based on neutrophils, a type of white blood cell. These Nutribots successfully navigated the blood-brain barrier in mice to deliver cancer-fighting drugs. One of the biggest hurdles in treating brain diseases is getting medication past the blood-brain barrier, which acts as a protective shield. While researchers have tried using natural cells like neutrophils and macrophages to carry drugs, these methods have been limited by slow delivery. To improve speed and control, scientists engineered Nutribots with magnetic material, allowing them to be guided with external magnetic fields. These micro-robots proved incredibly fast and maneuverable, reaching speeds 50 times greater than natural neutrophils. In lab tests, researchers demonstrated the Nutribot's ability to penetrate the blood-brain barrier and reach tumor cells. The Nutribots released their drug payload in response to inflammation signals, effectively targeting the tumor. Mice treated with Nutribots showed significantly longer survival rates. This innovative approach offers a promising new avenue for treating brain tumors and other neurological conditions that are difficult to reach with conventional drug delivery methods. Future research will focus on refining the design of these micro-robots, improving their drug-carrying capacity, and testing their safety and efficacy in larger animals before eventually moving to clinical trials in humans. When we get sick, what prevents the countless bacteria and viruses that invade our bodies from reaching our brain? As a matter of fact, the brain has a protective layer called the blood-brain barrier, or BBB, which is a barricade of cells between the blood vessels and the brain. This layer keeps the brain safe from disease-causing pathogens, such as bacteria and viruses and other toxins that may be present in your blood. The blood-brain barrier is a highly selective, permeable barrier that controls what gets through the bloodstream into the brain and what does not. The BBB is designed to keep harmful molecules from reaching the brain, while essential molecules such as glucose, oxygen, and water can easily pass through. Blood vessels in the brain have specialized endothelial cells that are packed very tightly, forming tight junctions. Small molecules leave the blood and enter the brain tissue by sneaking through these tight junctions. However, the BBB poses one of the major challenges for drug delivery to the brain, as it blocks almost 95% of medicine delivered orally or intravenously. To treat neurological diseases, such as Alzheimer's and Parkinson's, researchers are developing techniques to bypass this barrier. One method is the Trojan horse approach, where a therapeutic drug is fused to a molecule capable of crossing the barrier. This could involve combining the drug with antibodies that bind to receptors on the barrier's surface, essentially tricking the BBB into letting the drug pass. Another strategy utilizes engineered viruses to carry therapeutic agents directly into brain cells. Similarly, focused ultrasound could be used to temporarily open the BBB, allowing drugs to